Hi guys, I'm Aditya, a high school innovator that's currently researching computational biology and artificial intelligence. This video goes a little bit into how I use the visualization tool PyMOL for examining 3D representations of the 1NOW, the 1PCN, the 1MJE, and the 1MIU structures, and it looks into detail about various protein-protein and protein-DNA interactions. Before we start, here are a few terms that you have to know. PyMOL is an open-source molecular visualization system that is used commonly in molecular biology. To get a sense for the models that are needed for PyMOL, NCBI VAST, a service that allows you to compare a 3D structure against other structures, and PDB, an open source database of various biological macromolecule structures for research, were used. Lastly, BRCA2 was chosen because mutations of this protein have led to several forms of cancers, and this protein also helps the regulation of DNA damage. So what are we waiting for? Let's get started on how I did the project. So I just hopped on the protein data bank and I just searched up BRCA2 over here as a query, and here are all the related structures that we got. So let me just click on, oh look, 1NOW, we just looked at that before. So if I click on this, it says that it's a crystal structure of an RAD51, BRCA2, BRC repeat complex. That all doesn't matter, all we care about is that this is part of BRCA2. If we scroll all the way down to macromolecules, and this is the different macromolecules inside of the protein, we can see that there's a breast cancer type 2 susceptibility protein, which means this is part of the BRCA2 protein. And that's really important because we want to know that this is part of breast cancer, because this is a specific thing that I want to research, at least in my research. And the really cool thing is we can also look at the specific amino acid code, which allows us to do computational analysis on this. Now, after we got all this, let's just download the files over here. I'm going to download it as a PDB format because we're going to be looking this from the protein data bank, and this is also going to allow us to easily transfer it over to PyCharm. And let's just hop over to here. So I just clicked on another random thing that I was of the 4 structures that I was looking at. 1MIU, this is the structure of the BRCA2 DSS1 complex, and the DSS1 doesn't matter that much. All we care about this is part of BRCA2. And we can see that there's 24 related structures, and the most related ones are 1MJE and 1IYJ. 1MJE, again, is something that I was planning on researching, and has 95% sequence identity, which means that it's really related. And it has 592 aligned residues, which means that it's very, very similar. As you can see, these are 78 and only have 14% sequence identity after only 2 of these selected structures, which means that this is very rare. So we know that these are very highly related to each other, which means that they're both probably likely breast cancer susceptibility proteins. Let's hop on over to PyMOL right now. I've actually preloaded all of the PDB files for us to look at, so let me just push one of them over right now. Let's look at the 1NOW structure, for instance. As you can see, it's very nicely coordinated. Let's just move this to element by chain. So, as you can see, there's 2 different sections over here. There's this blue section, and then there's this green section. So what they are is different sections. And in these specific sections, we can even select certain parts of this section. And as you can see, if you can see that thing that's moving, that's actually the selected regions inside of the protein. And that's actually pretty incredible because we can see which specific amino acids inside of this structure are actually there. Another thing that we can do is also calculate the distance. So if we go to display, so if you go to wizard and then measurement, we can actually find the distance between 2 different atoms. So let me just click this atom, for instance, and this atom. And if I scroll in over here, you can see that it's 17.3 right over there. And that's pretty amazing that we can actually look at different distances inside of this protein. And another thing that I want to show you over here is the sequence window. So the sequence window is similar to what I was showing you before. It has a list of all of the different amino acid codes, and it allows us to make a lot of computational power between these. These zeros just simply represent that since there are a lot of mutations inside of this protein, that means that these are just canceled amino acids, and they don't have information on that. Let's switch over to another protein, for instance. So let's just look at the MIU protein. Okay, so there's 2 proteins over here. As you can clearly see, the MIU protein is definitely bigger. So let's just delete this object. Okay, here it is, and we have this measurement over here. I'm gonna get rid of this too. Okay, so we have this protein again. Again, we're gonna color by chain. Okay, as you can see, there's a blue part and then there's a green part. We can do a variety of different things. We can show this as lines, non-bounded, which I just clicked over here. I'm gonna move this back to cartoon mode. And it's really cool because now we have two representations. We have the cartoon mode, and we also have all these different lines, which allows us to see the specific structure and how the different nucleotides are paired with one another. We also have hiding. We also have labeling, but these aren't that as important for us at this point. What we want to show you over here is if we just click over this, we can actually select certain parts of the thing, and if we click red for instance, we can actually make one of these a red atom, and we can actually look at them. And let me just color this by chain similar how we did it before. I think it might have gotten deleted. And we can now see that there is a blue section and also a green section. So I hope this is a really nice introduction to kind of PyMOL and how I did visualization of structures. Look below in the thumbnail if you want to see a detailed article about all the different things I did in PyMOL. But I wanted to make this a really quick overview just to give you guys a little heads up on how PyMOL works. Hope this was helpful. Bye guys, see you next time.